rest of this hour, how much do genes influence behavior? We've all heard of the nature versus nurture debate. It's been going on for generations. How much of our behavior is due to our genes? How much due to our environment? Can you really separate the two? And when the Human Genome Project came along, the race was on to find the genes thought responsible for many illnesses, scattered in with the news reports of genes linked to cancer or Alzheimer's, or tantalizing reports of maybe there's a fat gene, or an aggression gene, or a monogamy gene, or even a gay gene, genes that seem to influence or control behavior. The discoveries seem to suggest that a simple tweak might be all that was needed to tame a savage beast or keep a philanderer home at night. Just a little tweak of the genes. Well, could changing behavior really be as easy as changing a single gene? Lots of people were and are skeptical. But now a new study this month makes the suggestion that at least in fruit flies, a single gene can change behavior. Scientists reported this month in the journal Cell that changing just one gene in a fly altered its sexual behavior. And in another experiment reported in the journal Science, researchers manipulated the DNA of the prairie vole, causing some of the animals to become more social. So how conclusive is this evidence for a gene-behavior link? Joining me now to talk more about those studies and the role genes play in the behavior of flies, voles, maybe even humans, is my guest, Gene Robinson, Director of the Neuroscience Program at the University of Illinois at Urbana-Champaign, and the new elect to the National Academy of Sciences. Well, congratulations, Dr.
Thank you very much. Nice to be here, Ira.
You're welcome. Let's talk about the study, recent studies. Let's talk about the fruit fly study. That seems to have really shaken up a lot of people.
Well, sure. It's a really exciting study. The researchers have been focusing on a gene called the fruitless gene, which has been the subject of previous studies that really have linked it with the control of sexual behavior in the fruit fly. This is a gene that is in the category that the technical term for it is transcription factor. A more general description is that this is a gene that orchestrates and controls the production of many proteins. So right away we were forced to confront the idea that we can manipulate a single gene, but by no means is there a simple relationship between a single gene and a complex behavior. There's a lot of intervening biology.
Because of the proteins. Let's go back and decipher what happened in that study first. The researchers put that gene that codes for the male form of the protein, and they put it in a female fly, correct?
That's right.
And they found that the females were acting more like males.
Correct.
In what ways?
Well, in their courtship behavior. So males will court females, and so females that were given a male version of the gene were then acting more like males, and vice versa.
So that tells us that if they have that version of the gene, they'll be sexually attracted to females no matter what sex they are.
That's right. And if you look carefully at the data, what you see is that there will be a probabilistic change. There will be an increase in the likelihood of responding in that way. It's by no means all or nothing.
Mm-hmm. And they had a whole bunch of different behaviors, tapping, singing, wing vibrating, all from that single gene change.
Yes, those are all part of the courtship behavior of the fruit fly, and so it would make sense that they would be connected in that way.
Mm-hmm. Did they put the female version of the gene into the male?
Yes.
And what did they get? Did they get similar reaction?
Yes, they get a similar reaction. Yep, exactly.
Mm-hmm. Let's talk about the vole study, too. That seems a little bit more complicated. What were the researchers working with? Vasopressin receptor gene.
That's right. Yeah, so it's a gene that encodes a protein that then allows a particular hormone to do its thing in the brain, to work in the brain. So it takes the receptor for the hormone to work.
Mm-hmm. And tell us what happened.
So what was done in this study was there was the observation made that there's variation in the length of a part of the genome that's just before where this gene is located on the genome. And variation in the length of a particular region of the genome was correlated with differences in the tendency to have social behavior, a particular form of social behavior, which was a kind of a bonding type behavior in the laboratory, and then manipulations of that region of the genome could cause changes in behavior.
Whether the vole was a good parent or not depended on that form of gene.
Yeah, they didn't do any real parenting assays, but they did the kind of bonding that you would expect would have that kind of repercussion down the line. They looked at how much time a male spends with a female that it has mated with previously. And males with the longer version of that piece of DNA spent more time with the female that they mated with previously than did males with the shorter version, suggesting that they would be bonding with that individual and perhaps be more monogamous.
What I also found interesting about the vole study was that the genetic differences were found in what we used to call junk DNA.
Yes, you're right. That's interesting. It's an old term, and it's quickly going out of fashion as discoveries that were fueled by the Human Genome Project and genome projects of other species have been leading to just marvelous discoveries, finding all sorts of roles for pieces of DNA that are in between the genes, the so-called junk DNA, that are now found to affect when a gene is expressed, where a gene is expressed, and how much of the gene is expressed, how active is the gene expression, how much of the protein product is produced. So we're really learning a lot about these other areas of the genome that don't encode proteins. And they're important.
Maybe not junk after all.
Exactly. Certainly the work on the voles and the fruit flies, these papers that we're talking about today, are the culmination of very nice research programs that have been done on these genes for some time.
And so how do scientists now move this forward to a next step? Where do you take this and move on to further tests and other studies?
Well, one of the big questions in the fruitless story is since the fruitless encodes a protein which is, as I said before, a transcription factor orchestrating other proteins, the question is, who is fruitless talking with? What proteins are interacting with fruitless? How do they work together to create aspects of the fly nervous system that show these sex-specific differences? So that's a really big question, identifying the genes or the proteins that are involved with fruitless. That's something that's really very important.
And in the vole side?
In the vole side, well, there's a hint of a really interesting new direction at the very end of the paper, and that is looking at the DNA sequences of primates and looking for similarities and differences in this same region of the genome that was looked at for the vole, to get some ideas of whether there are strong differences, consistent differences that are species-specific there in lineages of primates, and then to sort of look at the differences in primate social behavior. So it's just something that was done at the very end of the paper just to sort of look like a beginning of something, and I think that could be a very interesting line of research. What we have now is because we have the genomes of multiple organisms being sequenced, we have the ability to look at regions of the genome that are otherwise very difficult to study, the regions that don't encode the genes themselves. And so we're, with our genome sequencing projects, we have the ability to look there and sort of develop a stronger comparative base, which can then lead to new experiments.
Thank you very much, Dr. Robinson.